I'm Eric Ronne, a Beckman Scholar and undergraduate researcher in the Sinha lab at UC Davis. My project seeks to characterize the underlying genetics involved in leaf development and morphology in two plant species of the genus Lepidium using transcriptomics methods. Leaves form the interface through which plants collect energy from sunlight and exchange gases with the environment. There is a huge amount of variation in leaf morphology across plant species, two such being Lepidium oleraceum and Lepidium hissipifolium. Hybrids of these two plants exhibit a wide variety of leaf structures. By employing a transcriptomics approach, I hope to analyze leaf development and morphology from a functional genetic standpoint. But first, I'd like to take a minute to describe what exactly transcriptomics is. Perhaps the easiest way to do this is to say what it's not. While a genome is the base pair sequence of an organism's DNA, a transcriptome captures only the RNA that is transcribed from that DNA, providing a picture of what genetic material is expressed, not just what is present. Transcriptomics, therefore, involves harvesting RNA from sample tissue, sending that material to and receiving sequence data from a sequencing facility, reconstituting the disjointed sequence data into the original RNA sequences, and then performing analyses using the end result. While transcriptomics, like genomics, has many useful applications, accurately reconstructing and working with transcripts using only sequences that are usually less than 100 base pairs long requires a vast quantity of reads, and processing such large data sets in turn requires vast quantities of computing power. However, due to continuing advancement in the field, the powerful set of tools presented by transcriptomics has become more accessible. Improvements at every step, from sequencing technology to specialized software to computing power, which I employ at the Texas Advanced Computing Center, have made transcriptomics an increasingly important experimental approach. Now that you have some background in transcriptomics, let's jump into the project. The two species that we are studying are sister species in the Lepidium genus, Lepidium oleraceum and Lepidium hissipifolium. As can be easily seen in these pictures, the two plants look quite different, and one exhibits simple leaf structure, while the other has much more complex leaves. But, despite their differences, these two plants can hybridize to produce viable offspring, which, after generations of self-fertilization, exhibit unique leaf morphologies all across the spectrum from simple to complex. In order to assess the genetics behind these leaf morphologies, we must first construct a picture of their parents as a reference. This process begins with the harvesting of RNA from plant tissue in the parental species, followed by RNA sequencing at a sequencing facility. This process provided us with 202 million raw reads, well over 10 billion base pairs of sequence data. Using a variety of tools and scripts, reads that were unusable because of their length, sequence quality, or incorrect tissue identifier were paired from our dataset. Now with a set of reads for each of the two parent species ready, the next step is the assembly of transcriptomes. A transcriptome is made up of contigs that correspond to the RNA transcripts present in each sample. These contigs, in turn, are constructed based on the alignment and overlap of mountains of short sequences to each other. Without any published genome or transcriptome, we assembled the transcriptomes de novo, meaning that each contig was built solely from our read data. These raw transcriptomes will serve as a basis for all of our other analyses. However, it is key to filter and validate these sets of contigs before moving forward. A tool called Express was used to remove the contigs with the lowest read support. Transdecoder is a tool that identifies base pair sequences at the beginning or end of sequences that correspond to start or stop elements expected to be in real transcripts. And finally, CD-HIT clusters highly similar contigs together in order to remove duplicate contigs that correspond to the same real transcript. As we filtered our transcriptomes, we used the published transcriptome for a related species, Arabidopsis thaliana, for annotation and validation. Using a tool called BLAST, we queried the Arabidopsis database using our constructed contigs, associating gene identities and descriptions from the database with them. From the beginning of our filtering process to the end, the percentage of our assembly that had hits in the Arabidopsis database increased, even as the number of contigs were pared down to less than 1/6 of their original quantities. 90% mapping to Arabidopsis is a very encouraging sign that our assemblies are of sufficiently high quality. In addition, although our two assemblies were performed using completely distinct sets of reads, L. hippifolium and L. oleraceum also show around 90% overlap with each other among their annotated contigs. The roughly 2,000 annotated contigs that are uniquely present in one species or the other are starting points for our investigation of the genetics that underlie the differing leaf morphologies of these two plants. Several intriguing genes, a few of which I have included here, are among these uniquely present annotated contigs. YAB5, for instance, is involved in activating leaf lamina growth. LSH3 is known to be involved in regulating leaf complexity. And these are just a few genes of many that will likely find a place in our ongoing analyses. Thus far, our two Lepidium species have been hybridized, producing hybrid lines with widely varying intermediate leaf morphologies. Harvesting RNA from the parental species provided us with the sequence data which, after filtering, form the basis for the assembly of a transcriptome for each parental species. And, after filtering and validating these assemblies, we've begun to annotate the contigs present and find the similarities and differences between the two species that will form the foundation for our further analyses. Now that we have a detailed and carefully filtered reference in the form of two parental transcriptomes for this two-species study system, we are ready to begin with further downstream analyses. We will annotate more of our transcriptomes using Go and GoSlim tags, use read mapping to assess expression levels, and find genes that exhibit large expression differences between species, and use expression data to find clusters of co-expressed genes. This research is ongoing, and we hope to use these methods and others to glean further information about the genetics underlying the leaf morphologies of our two parent species and their hybrids. I would like to thank the Arnold and Mabel Beckman Foundation for supporting this research. Also, a thank you to everyone in the Sinha lab, and especially to Ashish Ranjan and Nilima Sinha.